My name is Will Townsend, and we'll be looking at a paper by Mansueto and Armani and Associates entitled Transcription Factor EB Controls Metabolic Flexibility During Exercise. Before we look at this experiment in particular, let's get an idea of the context that we're in. This experiment looked at TFEB in muscle tissue. Now, muscle tissue is somewhat unique compared to other tissues in how it experiences a vast spectrum of stresses on its energetic metabolism. For instance, you can have tissue that is fairly starved but may be required to go through vigorous contractions, or you can have muscle tissue that has plenty of nutrients that may need to go through many contractions. Likewise, you can have either starved or well-fed muscle tissue that may be at rest. And in each scenario, you need to adapt to what is going on so that you can maintain homeostasis and adequate ATP levels. The adaptive nature of muscle metabolism is enabled through a wide variety of chemical pathways that are all interconnected and incredibly precise. For instance, if you think of muscular contractions, there's a wide web of chemical reactions that takes place inside the muscle cell that helps maintain ATP levels after the hydrolysis of the current storage of ATP. And that can range from the addition of new glucose transporters into the sarcolemma to the biogenesis of more mitochondria for more aerobic respiration. Some of those pathways involve transcription factor EB, which we'll call TFEB from here on out. TFEB is sequestered in the cytosol of the cell and thereby kept inactive by mTORC1 mediated phosphorylation. mTORC1's activity level is modulated by nutrient availability. In nutrient-depleted scenarios, mTORC's activity is decreased, leading to less phosphorylated TFEBs. In scenarios with available nutrients, mTORC1 is very active, and thus TFEB is fairly inactive. mTORC1 is not the only factor involved in modulating TFEB's activity. Calcineurin, whose active form requires increased calcium levels, dephosphorylates TFEB, thereby activating it. The dephosphorylation of TFEB enables it to pass through the nuclear membrane into the nucleus, where it can carry out its duty as a transcription factor, increasing transcription of certain genes. So we might hypothesize that TFEB's activity might be maximized in a scenario with high activity and low nutrient availability. Some things are already known about TFEB, such as the pathways previously described and its responsibility for lysosomal biogenesis, as well as autophagy in the cell. However, there are still many more things to be discovered. The purpose of this paper was to investigate TFEB's impact on energy metabolism in the muscle cell, with particular respect to glucose homeostasis and mitochondria. To do this, a population of mice was divided up into three groups: a control group, a TFEB overexpressing group, and a TFEB knockout group. These groups were further divided into sedentary and exercising mice, as well as PGC1 alpha knockout and PGC1 alpha expressing mice. So we can see three controlled variables: TFEB expression, exercise, and PGC1 expression. As we'll see in the results, some of the dependent variables were mitochondria quality and number, TFEB location and density, the expression of proteins downstream of TFEB, and cell metabolite concentrations. Some of the techniques used to gather data were electron microscopy in conjunction with immunostaining, fluorescent microscopy, Western blotting, euglycemic hyperinsulinic clamps, and chromatin immunoprecipitation. So let's move on to the results. There were approximately 400,000 figures and 3 million tables, so I picked the ones that seemed to get at the main thrust of the paper and excluded the other ones. Let's start with figure 1. Looking at pictures B, C, and D, we're looking at tissue slices from muscle from control, TFEB overexpressing, and TFEB knockout, respectively. Note the differences in mitochondrial density and mitochondrial quality. B, the control, shows normal levels of mitochondrial number and quality. C shows an overabundance of mitochondrial number, while the knockout shows a significant decrease in the mitochondrial quality. Looking down at graphs E through H, we can see the same thing communicated. Cells which overexpress TFEB showed a greater number and size of mitochondria, while cells that had TFEB knocked out greatly increased numbers of abnormal mitochondria. Looking now at supplemental figure 4B and D, looking at B in particular, we can see that the overexpression of TFEB resulted in both an increased number of mitochondria and increased mitochondrial size, and this is in spite of the knockout of PGC1 alpha. We can see that the overexpression of TFEB led to an increased expression of all these proteins, and in every case, this occurred in spite of a lack of PGC1 alpha. The white bars show mice that had normal levels of expression of TFEB, while the black bars show mice that had increased levels of expression of TFEB. The pairs on the left side of each graph show mice that had expression of PGC1 alpha, while the pairs on the right showed those that had a lack of PGC1 alpha. Figure S4A shows the same movement of TFEB from the cytoplasm to the nucleus, and this happens in spite of the lack of expression of PGC1 alpha. So PGC1 alpha is not required for TFEB's translocation. Moving on to figure 2A, we're looking at proteins involved in mitochondrial biogenesis as well as energetic function. The x-axis is showing us which protein the graph is specifying, while the y-axis is showing relative levels of expression. What's important to note here is that in every case, overexpression of TFEB led to an increase in the expression of these particular proteins, while knockout of TFEB did not necessarily lead to a decrease. Note especially on the far left side, the increase in TFEB also led to an increase in PGC1 alpha. Figure S2 and D, we're seeing similar results to the last one, where increased expression of TFEB, as exemplified in the top left, led to increased expression of all these other proteins involved in mitochondrial biogenesis. You'll note the familiar PGC1 alpha in the bottom left-hand corner. Other very important proteins to notice are TFAM, NRF1, and NRF2. Those are nuclear respiratory factors. We're going to switch gears a little bit now, and in figure 4, think about exercise and TFEB expression. In part A, looking at our electron microscopy immunostained images, we can see that the TFEB knockout tissue showed no signs of TFEB, and that makes sense. The wild type shows TFEB spread out fairly evenly throughout the cells with a little bit higher concentration towards the edges. And the wild type exercise one is the interesting image because it seems like TFEB has all but abandoned most of the cell and moved into those little tiny clumps. Well, those clumps are where the nucleus is, and if you think back to the beginning where we talked about pathways, that makes sense because exercise results in calcium coming into the cell, which leads to the dephosphorylation of TFEB, allowing it to move into the nucleus. That movement into the nucleus allows it to do its duty as a transcription factor, and if you move over to part C, you can see that in action, where exercise leads to the increased activity of all these downstream proteins from TFEB. Figure S4A shows the same movement of TFEB from the cytoplasm to the nucleus, and this happens in spite of the lack of expression of PGC1 alpha. So PGC1 alpha is not required for TFEB's translocation. Figure 6, we're switching gears a little bit. The white bars on the graphs show mice expressing normal levels of TFEB, while the black bars show TFEB overexpression. And on the bottom, you'll notice two pairs of each. There's wild type, which has expression of PGC1 alpha, and then there are PGC1 alpha lacking mice. Regardless of the expression of PGC1 alpha, overexpression of TFEB leads to increased expression of glucose transporters, which will enable a greater intake of glucose into the muscle cell in response to TFEB activation. Figure 5. We're looking at a variety of molecules involved in metabolism in muscle cells. Note especially in the top right-hand corner, lactate levels are significantly decreased both before and after exercise in mice that overexpress TFEB, as shown by the red bars. If we were to postulate why this might be, we would recall that an overexpression of TFEB leads to an increased mitochondrial size as well as increased mitochondrial number in those particular cells, and also an increase in the expression of proteins involved in aerobic respiration such as complexes 1 through 4 and citrate synthase. So these cells are going to have more ability to perform aerobic respiration and less need to produce lactate from anaerobic respiration. Note now part J, free fatty acid concentration inside the muscle cell drops significantly after exercise in TFEB knockout cells. Now this would make sense with the previous graphs L through N because it would seem that glucose has a decreased availability, and so the cell must now rely on free fatty acid for energy. In summary, we can see that TFEB is heavily involved in all things mitochondria, from biogenesis and the transcription of genes involved in that to function and size and number. TFEB is also involved in glucose homeostasis, as knocking out TFEB resulted in a type of insulin resistance where the cells did not take up as much glucose and had to rely on free fatty acids more for energy. And so we might say, as a sort of overarching statement and conclusion, that TFEB is actually a major player in energy homeostasis, as it is involved in mitochondrial biogenesis and effectivity, as well as glucose homeostasis. Previously, this was not known, as it was just thought that it was involved in lysosomal biogenesis and autophagy. Also, all these things are independent of PGC1 alpha, and this is significant because PGC1 alpha has been seen to be a key regulator of mitochondrial biogenesis and function. However, evidently there is some other pathway that TFEB can utilize, although it also does utilize the PGC1 alpha pathway as seen in some of the earlier graphs. However, more research will be needed to investigate these alternate pathways.